Thanks for the organizers for inviting me. It's a real honor and pleasure to be here and share with you some of the work that we're doing at NIST, and also give you an overview of the exciting field of microbiome. I'm probably going to struggle through this, as I picked up a upper respiratory something or another earlier this week, and I'm at the stage now where I'm starting to lose my voice. So I've been quiet all morning trying to reserve my vocal cords for this, but nevertheless, I'll jump right in. So Misha mentioned that I work at NIST, and I know a lot of people aren't familiar with NIST, so I feel obligated to introduce NIST. NIST is the National Institute of Standards and Technology, and we're a non-regulatory federal agency in the Department of Commerce. We're the oldest technical institute in the U.S. government, dating back to 1901. We have two large campuses, one located in Gaithersburg, Maryland, where I'm sitting right now, and also in Boulder, Colorado. Pictures of them are on the, as you can see. If you look closely, I bet you can guess which of these campuses is in Boulder. We're about 5,000 scientists and engineers strong, about $1 billion annual budget. And, you know, when I say Technical Institute and the Department of Commerce, sometimes I get some wonky sideways looks. Like, why does the Department of Commerce need a technical institute? Well, our mission is to promote U.S. innovation and industrial competitiveness by advancing measurement science standards and technology. So we really see industry and industrial competitiveness and development as, you know, sort of our stakeholder. A fun fact, actually, this is just something I learned last month when I was reading an article in Science about NIST. Every dollar of taxpayer money that's invested in NIST returns $9 to the U.S. economy, which I thought that was kind of nice to see. Your tax dollars hard at work. So in the Biosciences Division at NIST, we think a lot about the bioeconomy, and that brings me to why does NIST work in the microbiome space? And it's because there are hundreds of companies, large and small, that are developing products that target the human microbiome. And when I say large and small, I mean like big pharma that you can see here, like Pfizer, and, you know, small startups that may be just a few people who, you know, came from an academic lab where there was some exciting discovery that they felt had commercial potential and stepped out into the exciting world of entrepreneurship. So, you know, when I justify my existence at NIST, this is sort of the slide that I show and discuss the bioeconomic ramifications and implications of microbiome science. Excuse me. So, as the first speaker, I was asked to provide a general overview of what we're talking about today, the human microbiome, more specifically the gut microbiome. Excuse me. The human microbiome is defined as all the microbes that live in and on our bodies. And these include bacteria, of course, but also viruses, protists, fungi, and other little things. Fun fact, there are more microbial cells in and on your body than there are human cells. The exact ratio there has been hotly debated. It used to, you know, be thought it was like a 10-to-1. And then some said it was fewer, but I think the scientific community agrees that there's more microbial cells than human cells. And these are largely found in your gut. That's where a majority of your microbiome resides, but they're also in your skin and your urogenital tract and on and on. This is not a monoculture of bacteria. There are literally thousands of different species of bacteria that reside in and on your body. And in our gut, you know, you might have hundreds of different species of bacteria that form a resilient ecosystem. You know, these things aren't operating in silos. They are a community that work together as a community. And the community function and outcome, also known as maybe a phenotype, can impact your health. I think it's also interesting to note that the human microbiome contains about 3.3 million non-redundant microbial genes. And just for context, compare that to the human genome, your human genome, that contains about 22,000 genes. So many of these genes contain enzymes that, you know, transform biochemical processes. So you can imagine, you know, this extremely rich potential of biochemical transformations that can and do exist within your gut microbiome. Our microbiomes impact us metabolically, neurologically, immunologically, and hormonally, and that pretty much covers, end to end, everything happening in our bodies. And to make things more complicated than they have to be, every individual on Earth is very unique with respect to their microbiomes. And, in many cases, individuals, even closely related, husband-wife type relationships only share a small percentage of their microbiome, on the order of about 10%. Take that into the context that if you think about our human genomes and our relatedness as Homo sapiens, we're all about 99.9% related as Homo sapiens. But if you look at our microbiomes, we're only about 10% related. And this brings me to the last most poignant statement, is that the human microbiome plays an important role in human health and disease and represents an understudied and underappreciated target for new diagnostics and therapeutics. And maybe that statement's a little bit outdated, because I think now we know that it is we appreciate its implications in health and disease. And, all of those companies that I had mentioned in the previous slides, and academic labs, et cetera, those are the ones that are developing these clinical applications of the human microbiome. So to be sure, earlier today, when I was putting together some of these slides, I did a quick search on clinicaltrials.gov for microbiome keyword and found 3,400 studies. Some of these have been completed, some are enrolling, some are recruiting, some are ongoing, but 3,400 clinical trials that include the keyword microbiome. And if you just do a quick scan down the list here, and this is just 8 out of 3,400, you can see, the microbiome of pancreatic cancer, and, the vaginal microbiome, ocular microbiome, and immune system and dry eyes, and on and on and on and on. Yes, there is a lot of hype. I always get a little nervous when, you're standing in line at the grocery store checkout line and you're looking at the magazines, and on the cover of one of these magazines is something about your microbiome. And I think, gosh. So here's just one of those, a 16-week vegan diet can do wonders for your gut microbiome, when in fact, we don't really know what a good microbiome or a bad microbiome is. I know that's a little disheartening to hear, but it's true. So while we could debate whether a vegan diet is good for you or not, we really don't have any way to measure how good it is on our gut microbiomes. And this brings me to this Gartner hype cycle, which is something that kind of describes new technologies. And when a new technology comes out, not many people know about it, but then, the media catches on, and you get this peak of inflated expectations where everyone says this is the next huge thing and it's going to change the world. And then, reality sets in, and there's this trough of disillusionment, the slope of enlightenment. And then finally, we plateau off to some productivity. Where microbiome is on this curve, I don't know. I like to speculate, and I don't know, maybe we're on this side of the trough of disillusionment. Maybe we're still on this side of trough of disillusionment. I don't know. Lee or Janet, can someone confirm that you can see my pointer? I just want to make sure that I'm not pointing just from my own.
I cannot currently see your pointer.
Okay.
Oh, now I can see it. Yes, when you move.
Okay, I was pointing at the wrong screen, thinking that you all could see that. Thank you, Lee. So at any rate, I was just pointing out, I don't know if we're on this side of the trough of disillusionment or this side, but we're somewhere along the way. While there is a lot of hype, I only show that to point out there's also a lot of really, really good science behind this. So I think one of the biggest exciting discoveries with microbiome science in the last 10 years is the realization that the human gut microbiome modulates the efficacy and response to immunotherapies in cancer patients. And this discovery was published in 3 back-to-back papers in Science in 2018, where it was discovered by, simultaneously by 3 different groups looking at this. So, if you'd seen it once in a small group, you would say, maybe not. But this was a real discovery, and a really important one too, because these immunotherapies that are used in cancer patients are really life-saving treatments. And what they have found is that if you can modulate your gut microbiome or change it to a more compatible or healthy state, that the outcome of your immunotherapy treatment could be more positive. And this is in 2018, so this is 5 years ago. And a lot of the oncologists that are on these papers have sort of shifted a lot of their focus of their research to understanding this relationship between the gut microbiome and your response to an immunotherapy. And while the mechanism is not understood, it's generally recognized that it's an immunomodulatory effect. You know, our guts are full of immune cells. Our microbes are you know, interacting with those immune cells. And of course, immunotherapies by definition are immunomodulators. Okay, so I'm going to be talking a lot about human fecal material, poop, in the coming slides. And I just want to clarify, in case it's not clear for anyone, the reason I'm doing so is because when we talk about the gut microbiome and we want to do measurements on your gut microbiome, the way we do that is collect your fecal material. You know, it's not invasive. Everyone poops. Everyone, you know, sometimes most people poop multiple times a day. So it's a convenient media to use as a proxy for understanding what's going on in your gut. And you know, as I pointed out before, you know, in a gut fecal sample, you know, there's hundreds or thousands of species of bacteria, and everyone is very different. And this brings me to fecal microbiome transplants. These have been in the news a lot. Some people say they're new, but in fact, they go back to, you know, ancient civilizations. You know, fecal microbiome transplants recently in the last 10 or 15 years have really come on the scene because of their demonstrations of really high efficacy at treating recurring and primary Clostridium difficile. You know, Clostridium difficile infections are a horrible disease that affect, you know, hundreds of thousands of people in the US every year, more around the world, and many young, healthy people die of C. diff every year. The standard of care used to be treatment with antibiotics, which is kind of counterintuitive if you think about what's happening with the gut microbiome. But now the standard of care is emerging as an FMT. So if you've got this nasty Clostridium difficile infection in your gut, you repopulate it with a fresh microbiome, that is, you know, fecal material, and it's highly efficacious. So it works well for Clostridium difficile, and you know, it's not a leap of faith to say, well, let's try it for other indications, other pathologies. And a quick search of clinicaltrials.gov this morning for fecal transplant revealed 555 studies, and they are all over the place in terms of indications. So hematopoietic stem cell neoplasms, treatment of obesity, treatment of irritable bowel disease and Clostridium difficile, of course, extreme drug-resistant bacteria, mental disorders like bipolar disorder and Tourette syndrome, Parkinson's disease. Of course, there's a lot of clinical trials ongoing to understand how gut microbiomes and FMTs modulate the efficacy of immunotherapies that I mentioned before. And even, you know, disorders like alcohol misuse and cirrhosis. You know, think about, you know, treating alcoholism with a FMT. I'm not saying it works. I'm just saying that these are the, you know, applications where things are being explored. In fact, many of these probably won't work. But it's you know, it's another you know, tool in our belt. So this was big news 8 months ago, I guess, in November of 6 months ago. In November of 22, the FDA approved the first microbiome targeted therapeutic. It's known as Rebyota. As you can see in this bag, it is a FMT-like material. There's still some confusion and some debate in the field as to whether this is an FMT or isn't an FMT. Nevertheless, if you look at the label that, you know, this material comes in, it says Rebyota is manufactured from human fecal material, and Rebyota contains live microbes. So you know, it doesn't say on here necessarily that it is a fecal microbiome transplant. Yes. If you think about the manufacturing of this, this is derived from human fecal material. So, you know, just as you might expect, they have screened donors that come into their facility every day and provide their bowel movement donations that undergo some processing in their facilities. And it's, you know, some proprietary processing that produces this standardized output drug. So thinking about quality control of biomanufacturing of either Reb Yoda, or I've got FMT here. And again, we're not quite sure if Reb Yoda is an FMT or if it's something else, but what we need to know in order to produce this and use this, in a safe and effective way, we need to understand what microorganisms are present in the material. That's, these are analytical measurements that are necessary. What are the relative quantities of the different microorganisms? Again, there may be hundreds of different species. So what are their relative abundances? Are they all equally abundant? Probably not. Does it matter that some are more abundant than others? Probably. Are they viable? So some of these organisms are viable, some aren't, some die off, some are anaerobic, that is that they die in the presence of oxygen. So are there any contaminants? That's an interesting question to think about. There's hundreds of different species are in these products, but are there any organisms that aren't supposed to be there that might constitute a safety issue or have an adverse event, like a pathogen or some environmental isolate? How stable is this material? What's the shelf life, the viability? Do the cells die off over time, as you might expect they do? How do you even measure potency? Any drug that's manufactured needs an in vitro potency assay. What is potency for a therapeutic like this that we don't even understand the mechanism of action? Similarly, what's a dose? How do you define a dose? Is it a certain number of organisms? And going back to this human donor manufacturing process, it can't be 100% reproducible in terms of batch to batch reproducibility. So what is the variability of batch to batch reproducibility? I mentioned this because I probably didn't point out enough, but NIST is a measurement science institute, a metrology institute. So we don't do clinical trials. We're not a public health agency. We focus on measurement science. So it's exactly these kinds of questions that we geek out on and we work on. And, we work with biopharmaceutical companies a lot. This is how I got to know Yvonne Navor, our next speaker, who works on quality control and biomanufacturing of microbiome products. So if, the way that Reb Yoda is used clinically, if you have a cohort of patients with Clostridium difficile, and by the way, I don't know if I mentioned it, but Reb Yoda, it is used to treat recurring Clostridium difficile. So if you have, patients that have Clostridium difficile, you treat them with Reb Yoda, this live biotherapeutic product, and, presumably it restores their healthy gut microbiome and it cures their disease. And, presumably you'd want to analyze the patients before and after. And when I say analyze, how does the therapy change the patient's gut microbiome? Can the therapeutic microbes be detected in the gut? So, if I'm a patient, I've got some, gut microbes of my own, and then you're going to introduce more via this therapeutic modality. And, as a researcher, you might want to know, can I detect the therapeutic microbes over my normal gut flora? It's interesting that the field has sort of merged on using the word engraftment. We hear that a lot with tissue transplants, but it's not that different. That you're taking cell tissue material and putting from one patient to another. So you might ask, how long or how stable are these therapeutic microbes in the gut? And again, all these measurements are performed on fecal samples. So there's magic in poop. If I haven't already convinced you, poop can be highly efficacious at treating disease. However, we know that some poop works and some doesn't. So super poop, these companies that prepare FMT materials, they have some screening where they find donors that have, efficacious poop. Sometimes they're called super poopers. But we don't really know what the right poop looks like. That is, we can't measure one fecal sample and measure another one and say this one is right and this one's wrong. We don't know. We don't know what the thing or things are that we can measure to determine if an FMT is efficacious or not, or if it will be efficacious. To make things, a few orders of magnitude more complicated, the gut microbiome has been described as the most complex ecosystem on Earth. And remember, it is an ecosystem. You can't just add bugs willy-nilly. These things have evolved to live together and work together. It's hard to measure. The clinical applications of the human microbiome are therapeutics and diagnostics. And I'm going to talk quickly about microbiome diagnostics. And I'm sure most of you on this call are familiar with the 23andMe model, where if you want to get your personal genome analyzed from 23andMe, you sign up on their website. You pay them to have a kit sent to your house. They send you a kit. You open the kit. You provide some saliva or buccal swabs, and send it back, and they analyze it, and they send you a report about your personal microbiome. That same model is being used for direct-to-consumer gut microbiome testing. And this is a article that really emphasizes some of the problems and challenges around this. So this science reporter, her name is Tina Hirschman. She wanted to look into this as a science reporter, and she identified two companies, American Gut and MuBiome, that offer these direct consumer testing. So she took one of her fecal samples and she split it in half, and she sent half to MuBiome and the other half to American Gut. And as she reports in her article, I asked two different companies to analyze my gut microbiome, and they gave nearly opposite results with respect to the major phyla, bacteria, in duplicate samples. That is, one fecal sample. So being a science writer, she did her due diligence. She reached out to the companies and asked and told them, look, here's what I got, here's what I saw, and they're contradictory. And there's a lot of interesting quotes here. So one of the microbiome experts in the field commented that the lack of reproducibility between studies is frustrating. It seems like cowboy country. We need to have some kind of order. Another blogger who's a bioinformatician, he asked her for the raw data, the raw sequence data the company generated. He then analyzed that raw sequence data using his tools, and he got yet a third answer. So, but DNA extraction is not the only thing that could go wrong. It seems that every step in the process, from how you collect the samples through the computer programs used to analyze the data, is the potential culprit. All sorts of unlikely things are possible, and finding which one is true is difficult. So these are very NIST statements. NIST is a measurement science institute. We think about bias, analytical performance of measurements, and things like this. So this was probably a 9-year-old article. I can tell you, though, that the number of direct-to-consumer microbiome testing companies has grown considerably since this article came out 9 years ago, and I don't think the measurements have improved proportionally. So I'm going to talk about Metagenomics 101. This is really the lifeblood of what we do in my group at NIST. It's a very powerful tool. So if you have a complex sample, like a fecal sample or a soil sample, where you have hundreds or thousands of different species of microbes all in one sample, and you want to know what are the organisms, who are they, and what are they doing in this sample. What you do in a metagenomic measurement is you just simply do a DNA extraction. And remember, all life on Earth is nucleic acid based. So every organism on Earth is going to have either a nucleic acid element to it, which we can use to identify them. So after you extract DNA, you just simply put it on a sequencing instrument, and you sequence all that DNA, and you get a GATC type of sequence output. And from that sequence data, we can identify, based on the genetic signatures of the microbes from reference databases that we know, we can go back and say, okay, now we know who's in the sample, and we know what their relative abundance is. And this really wasn't available to us 20 years ago, because DNA sequencing technology was a lot more cumbersome, and it was expensive, and it was low throughput. But now DNA sequencing technology has been democratized, and you can do a microbial genome for like $5 now, and you can do a human genome for like $500. It's crazy. It's outpaced Moore's law. So that's just allowed us to really, leverage and utilize next-gen sequencing technology for metagenomics. The previous slide I showed you was sort of the 101 metagenomics, but this is the more realistic measurement workflow. I'll just point out, I like to think of metagenomics as the 21st century microscope, because just like the microscope did in the 17th century, it allowed us to see things that we couldn't see before. And that's exactly what metagenomics does for us. We can see things now that we couldn't see before. Instead of directly peering at them through a microscope, now we see their DNA signatures, which is just as good, almost as good. I don't know, maybe better. any rate, in the workflow, you've got your sample collection and storage. As I mentioned, you extract DNA. Then you're going to have to do some molecular manipulations of that DNA to make it compatible with your sequencing instrument. Then you put it on your sequencing instrument, and you generate the data. The data comes off, and you're going to do some filtering and trimming of that data. And then you have to do some statistical analysis and reporting. And another way of showing this workflow is using these Ishikawa diagrams, or sometimes known as fishbone diagrams. But it's just a way of showing, there's a multitude of methodological variables that go into this. For example, DNA extraction, there's dozens, hundreds of different ways to extract DNA from a sample. None of them are 100% efficient across all cell types, and they will all give you a subsampled representation of what your original sample was. Moving on to the next step, library preparation. There's dozens of ways to prepare a library for next-gen sequencing. They'll all have their internal strengths and weaknesses and biases. There's half a dozen sequencing technologies on the market that have their own strengths and weaknesses. And the point is, and this is a statement I make at least 15 times a day here at NIST, that there's bias in every step of the measurement process, from how you collect and store your sample to how you analyze your data and everything in between. So at NIST, we are, let me see if I can get rid of that. Okay. NIST, as I mentioned, is a metrology institute, and one of the things that we're probably known most well for is our development of reference materials. So if you come into our headquarters building and walk through the lobby, there's a showcase, and you'll see this display that displays some NIST reference materials that have been developed over the last 100 years. You can see that we have standard reference cigarettes, we have standard reference peanut butter. Here's a meat homogenate, sulfur and diesel fuel oil. Over here, we've got industrial sludge and domestic sludge. I don't really know the difference between them. And then we've got Portland cement. I don't know what's special about Portland. Some of these might seem silly, like, why do you need cigarettes and why do you need peanut butter? That's ridiculous. But I promise you, every one of these has a very important industrial application. On average, it takes NIST about 5 years to produce one of these reference materials, and not just 5 years, but a small team of PhDs about 5 years. So we don't commit to this lightly. We don't just like willy-nilly go out and say, hey, let's make a peanut butter standard. Like, there needs to be a really pressing industrial need for one of these materials before we'll commit to developing it. And this brings me to our next new NIST reference material, human fecal material. So this is something my group started about 4 years ago. And as I said, it takes about 5 years to produce one of these materials, and we're about a year out from having this ready. Here is some of the manufacturing processes that we use to make large quantities of homogenized fecal material. Ninja blending is an integral part of this process. The material needs to be homogenized, and excuse me, human fecal material is inherently heterogeneous. So more on the manufacturing of this material. We decided early on that for this NIST standard, we wanted, pardon the pun, 2 flavors of fecal material. So we rationalized that diet had the biggest impact on your gut microbiome. So to have 2 different types of human fecal material, we decided we would collect from 2 different cohorts that differ based on those diets. And as you can see here, those 2 cohorts are omnivores and vegetarians. So we recruited these 2 cohorts. We asked them to provide bowel movement samples, sometimes multiple bowel movements per donor. We pooled those bowel movements, did the homogenization using the old Ninja blenders. And then we produced about 5,000 aliquots of each, and each of these has about 1 mil in it. And we've spent a couple of years understanding how to stabilize this material, because this material not only needs to be homogeneous, it needs to be stable. It's no good to us if we manufacture it and put it in the freezer, and next year it's completely different than it was this year. The point of a reference material is that you can measure it today and you can measure it a year from now, and you get the same answer. So homogeneity and stability is really what it's all about when you're producing a reference material, and we've honed in on storage of this material at -80. The only additive here is pure water. We did play with some storage buffers and additives and adulterants, and we found that they all had limitations that were harmful to this material. So we decided just to go the neat route, neat being just water. So this is real, pure human fecal material diluted in water. I mentioned that human fecal material is the most complex. It's been described as the most complex material, biological material on Earth. I can't vouch that it is because I haven't sampled every biological material on Earth. But just some supporting facts here behind that. Human fecal material has been observed to contain up to 10 to the 12th microbial cells per gram of material. That's the highest cellular density of any known ecosystem on Earth. That is, scouring the Earth, we haven't found any other microbial ecosystems with a cellular density that high. So they're really packing the cells in there. Rule of thumb is that there's about 10 viral particles per microbe. So there's, an order of magnitude more viral particles per gram of fecal material. Think about the small molecule metabolites here. I'm saying thousands, but it's probably more like thousands of thousands, millions. Other biomolecules, proteins, RNA, lipids, all of these are very relevant, important. Human fecal material often contains human cells that slough off from your epithelial cell lining and your intestinal cell wall. You eat food that comes from plant and animals, and you will find plant and animal DNA and materials in your fecal material. So that's a part of it. And the reason I point this out is because this goes back to, what I mentioned about how there's super poop and some poop is efficacious, some isn't. We don't know the thing yet that makes some poop efficacious and some isn't, but whatever it is, it's something in this list. And it's challenging to measure these things. So every one of these things is a potential biomarker with clinical relevance. So, going back to our fecal standard, we've produced thousands of aliquots of a homogenized fecal material. And now what we're doing is multi-omic analysis. So metagenomic analysis that I mentioned, of course, that tells us the microbial taxa that are present and their relative abundance, but also a flow cytometry, a cell measurement techniques that shows us the cellular morphologies of the different types of cells present. Good old-fashioned Petri dish plating that shows us the different morphologies of the different types of cells that are present. Metabolomic analyses, mass spec-based metabolomic analysis shows us the small molecule metabolites present. We can also do metabolomics, and we do metabolomics using NMR-based techniques with some of the data shown here. Metatranscriptomics or gene expression. So, those cells that are in this fecal material have, some expression profile, and we can measure what that gene expression profile is using metatranscriptomics. Virome, I mentioned there's about 10 viral particles per microbial cell on average, and we can measure those viral materials. Lipidomics are very important biomolecules that have clinical implications, and metaproteomics as well. So when I say multi-omic analyses, this is what I mean. We're throwing the kitchen sink at this stuff, measuring every relevant marker that we can do. Okay, so I mentioned that this is, it's in the works. We've been working on it for about 4 years, and we expect availability late 2023. And I feel safe saying that this will be the most well-characterized human fecal standard on Earth. Let me close that real quick. Okay. So I'm going to shift gears real quick. I'm also noticing my time. And I'm going to talk about probiotics because if you all are clinicians or involved in human health and microbiome, this is a hot topic. So probiotics really come in 2 flavors. You can buy these, like CVS, the pharma companies over the counter, over the shelf. These are like purified bacterial strains that come in capsules, and you take it like a vitamin or a pill. And then the old school probiotics where these derive from are fermented foods, so kimchi and yogurts and cabbage and excuse me, on and on. So probiotics are not drugs, and I mean that in a regulatory sense. So the FDA has very clear guidance on what constitutes a drug and what doesn't constitute a drug. So probiotics, of course, contain live microbes, but these are dietary supplements according to the FDA. As dietary supplements, they're regulated by the Center for Food Safety, and the Center for Food Safety regulates them using a framework like that of vitamins. So think of the regulation of vitamin C or something like that. And that's how the FDA considers that. Since these are not drugs, the manufacturers cannot make medical claims. Even if there's clinical trial data that have demonstrated efficacy, you still can't make medical claims. So they make generic claims like friendly bacteria for your digestive system. That's what this one says. Okay, so back to the old clinicaltrials.gov. I did a search for probiotics, 2,124 studies using probiotics. And as you can see, they're all over the place. Here's one for HPV, exercise performance, non-alcoholic fatty liver disease, on and on. So one of the questions that I get, and it's worth mentioning, do probiotics really work? And you know, there are many clinical trials that have been performed in an attempt to demonstrate the efficacy of probiotics for treating myriad diseases. When I say myriad diseases, I mean all of these different types of things you see here. And you know, this question, do probiotics really work? It's such a generic question. Work for what? What probiotic? You know, there's many different types of probiotics, many different disease indications. Here's a 2018 systematic review and meta-analysis where they looked at, you know, a couple of dozen clinical studies that looked at different indications. So I apologize for the resolution of this, but this one on top is pediatric antibiotic-associated diarrhea. Here is adult antibiotic-associated diarrhea, Crohn's disease, Clostridium difficile infection, nosocomial infections, traveler's diarrhea. And you can see what they're showing here is whether or not there was a positive outcome. Was the probiotic, did it appear to be efficacious in these different clinical studies? And in some case, there appeared to be some efficacy. In some case, there wasn't. And I stole a couple of slides from a colleague, and I just want to show you some of these data. Sometimes, you know, even though statistics says it's significant, it's still a little hard to see. So here's, you know, using a probiotic, Lactobacillus GG for traveler's diarrhea. And, you know, here they're saying that, you know, the occurrence of traveler's diarrhea was about 5% in kids who received the probiotic and 8% in kids that didn't. And they say that's statistically significant. I'll leave it up to you whether you think that's significant. Here's a similar study where they looked at, you know, the probiotic Lactobacillus reuteri in acute diarrhea in children. The placebo is in blue. The probiotic is in yellow. And again, you know, you kind of squint and look sideways. You say, oh, yeah, okay, that seems to work. And in some of these, you know, I just highlight in yellow at the very bottom, you know, they just point out, hey, these differences were not statistically significant and there's no benefit of probiotics. So back to that question, do probiotics work? Sometimes. It depends on the probiotic. It depends on, you know, the indication, how they're treated. I just want to call out quickly Dr. Dan Merenstein. I don't know Dan personally, but I've heard him speak at a few conferences and I've read his papers. He's a pediatrician at Georgetown University, right down the street, of course. And he does a lot of clinical studies using probiotics, namely to treat antibiotic-associated diarrhea in children. And he's had a lot of success doing that. So I just want to point that out. If you're a clinician and you're thinking about probiotics for your patient, reach out to Dan. I'm sure he'd be happy to chat with you, or maybe not. As I said, I don't know Dan, but he's doing a lot of really great work, and he's a recognized expert and leader in this field. Many clinical studies fail or are not reproducible because of the poor quality of manufactured probiotics. So this is the dirty underbelly of the world of probiotics, and that is manufacturing quality. I'm sure Yvonne will speak more eloquently on this, but specifically, if you're taking a probiotic and it says something on the label on the side of the bottle, you know, is what is written on the side of the bottle on the label, is it what they say it is? Is it the right strain of bacteria genetically? And I can tell you from some personal experience in sequencing some probiotics that we bought off the shelf at drugstores, sometimes they're not. Purity. Are there any other organisms in there or chemical contaminants that shouldn't be in there? Are they alive? You know, presumably these bugs need to be alive to be efficacious, but during the manufacturing processes, they undergo these mechanically harmful processes like lyophilization and spray drying that can kill the bacteria. So, you know, how effective are they if they're all dead by the time you get them manufactured? What is a dose? They're typically on the side of a probiotic bottle, you might see 10 to the 9 live cultures or something like that. We've looked at that in our labs, and sometimes, they say it's 10 to the 9, but we get 10 to the 5. They're off by 4 orders of magnitude. Stability, the shelf life, how long do they stay alive? And batch to batch reproducibility. Again, if you're doing a clinical trial from a batch of probiotic produced today, and then you want to repeat it tomorrow, and the batch that you use tomorrow is produced differently, you're going to get a different clinical outcome. I'm part of a USP probiotics panel that formed about 5 or 6 years ago, and it was specifically formed to address this problem in the industry that is around quality of probiotics. So what this USP probiotics expert panel is doing is developing quality standards for the probiotic industry. And, a lot of times you see third-party validations in, the drugstores and the vitamin aisle, and one of those is USP. It's got the USP seal, and USP is a third-party independent certification and quality assurance agency. So, in the not too distant future, hopefully you'll start seeing probiotics on the shelf that have a USP seal of approval. This USP panel, we've developed a new USP general chapter that is for manufacturers to follow this protocol and how to do it right. We've published in peer-reviewed journals about improving end-user trust in the quality of commercial probiotics. Here we discuss, third-party certifications, setting standards for identity, purity, and quantification of probiotics. We talk about some emerging methodologies, namely metagenomics, that can be used to assess quality. I see I'm at 43 minutes, and I was told to go for 45, so I'm going to finish up after this slide. So some take-home message for clinicians around probiotics. Read the literature. The average consumer, non-scientist, can only read the bottle, and the bottle is going to say improves gut health. They're probably not going to have the wherewithal to go to PubMed, find the peer-reviewed literature, and read about how that probiotic strain was studied clinically and whether it was efficacious or not. But you, as a clinician, as a scientist, you can do that. So ask, in the literature, has a rigorous clinical study been performed? Did they use the gold standard randomized double-blind placebo-controlled trial? And preferably, this trial was not done by the manufacturer because, well, I don't need to say why, but preferably it was done by an independent party like an academic. There are thousands of different probiotic products on the market, like different strains. They're all different genetically. Lactobacillus acidophilus is a typical species of probiotic, but there are, innumerable different strains that are genetically different and therefore metabolically different and unique, and presumably they will all have different clinical outcomes. And to complicate that, some of the probiotics are blends of multiple or many strains. Because I'm in this field, when I go to CVS, I go to the probiotic aisle and I just start reading labels just out of curiosity. And some of these probiotics now will have 10 or 15 different strains of bacteria in them. So, is more better? I don't think there's any clinical evidence to say more is better. Also think about, fermented foods like yogurt, kimchi, sauerkraut that contain probiotics. People say I take probiotics. That could mean that I eat yogurt every day, and that's legit. But if you see some clinical outcome, is it due to the microbes, or is it just due to the food? You're increasing fiber consumption, et cetera. If you're eating kimchi, maybe the clinical improvement you see is just because you're consuming larger amounts of high fiber food. Partner with reputable companies if you want to do a clinical trial, a reputable probiotic manufacturer. And also, once you receive the product in-house, again, let's say you're going to do a clinical trial, you get all your probiotic material in-house, do an independent confirmation in-house to make sure that what you have is what's written on the label. Like you can sequence the genome of the bacteria and do some PCR and various things like that. Because again, there's no guarantee that what the manufacturer gives you is what's on the label. Excuse me. Okay, that's all I have. I just want to mention that NIST has very competitive and prestigious postdoctoral fellowship opportunities. They're run by the NRC for us. They're relatively high paying. That is that the postdoctoral fellows get paid $77,000 per year about. I think it might even be a little bit higher than that now for 2 years. Compare that to the NIH average right now that I think is about $55K. It also comes with a travel allowance, and our NRC postdoctoral fellows go on to do great things like win Nobel Prizes, literally, and become the NIST director. This is the incredibly talented and wonderful team that I've had the pleasure of working with. A very technically diverse group of people: analytical chemists, biomedical engineers, chemical engineers, microbiologists, etc. It takes a village. And that's all I have. And I think we're saving questions to the end.
Correct. We can move on.